Hello, Shashank sir, can we start?
Yes, sir.
Hello, am I audible?
Yes, sir.
Shashank sir, can we start?
Yes, sir.
Okay. Hello, am I audible and visible?
Yes, sir. Yes, sir.
Hello.
Yes, sir.
Okay, thank you. Sorry for the technical glitch. Good evening, everyone. On behalf of Microbiology Department, would like to welcome Dr. Anita, Principal, VES College of Arts, Science and Commerce. Invited speaker for today's webinar, Dr. P.V. Bhaskar, faculty members from various colleges and our esteemed alumni and dear student participants. After 4 successful webinars in past 3 months, Microbiology Department of VES College is happy to host the 5th webinar of Inspire Me series. This time again, our webinar focuses on a new and exciting area of microbiology, that is, as you all, the Department of Microbiology at VES College had started webinar under Inspire Me series in May 2020. These webinars are conducted with an objective of connecting you with scientists and research scholars that you receive the latest knowledge in those fields. We believe that today's is tomorrow's future. I now request our Vice Principal and Head of Department of Microbiology, Dr. Shantini Nair, to deliver. Over to you, Shantini ma'am.
Thank you so much, Malay. Never mind the technical glitches, we are getting used to it. We are most happy to host our 5th webinar of the series for Inspire Me webinar series. Honorable Principal of VS College of Arts, Science and Commerce, Dr. Anita Kanwar. Invited speaker for the webinar today, Dr. P.V. Bhaskar. All my colleagues from microbiology across Mumbai and the nation. All our esteemed alumni, all our former HODs and principal, and dear student participants from far and wide. Good evening, and again a very warm welcome to each one of you. A special welcome to our speaker for today, Dr. Bhaskar, who has joined us from Goa. The Microbiology Department of VS College of honestly takes a lot of pride in hosting the 5th webinar on polar microbiology, which has generated a huge interest amongst all students. We express our gratitude to all our student participants for their positive feedback for the previous webinars that we have conducted on arboviruses by Dr. Finny Varghese, on epigenetics by Dr. Subhojit Sen, on coronavirus cell interactions by Dr. Prajakta Oak, and the latest one on astrobiology by Dr. Rebecca Thombre. Some of them are prominent alumni. The speaker for today's webinar, Dr. P.V. Bhaskar, is a highly accomplished research scientist, and I must express my appreciation to the convener of this webinar, Dr. Shweta Patil, to help bring about this connect. In the brief interaction yesterday and prior to this webinar, he has enlightened us on a lot of exciting research opportunities in this field, which we are looking forward to learn from you, sir. On behalf of the department and entire college, I extend my greetings also to the pioneers of our department, Honorable former Prime Principal Dr. Jayashree Fadnis, Honorable former HODs Dr. Maitri Paul, Dr. Hema Varadarajan, and Dr. Nagesh Malik, as also to all our non-teaching staff members who have been the pillars of our microbiology department. I appreciate and acknowledge their role in building our team Micro, as has been projected in our departmental website recently launched, Microbytes, that has been so wonderfully designed and maintained by the convener of Microbytes, Mr. Suman Gangar, and his team peers of 2019-20 TYBC Microbiology batch. We are extremely proud of their sustained efforts at this wonderful initiative. Please do visit our website, and we would appreciate your comments and feedback on all the activities being projected in our website. I'm deeply indebted to VS management, especially our trustee in charge, Advocate Dr. Sri Lakshman Kanal, and to our extremely supportive and ever encouraging principal, Dr. Anita Kanwar, for all their unhindered motivation in conducting this series, and of course for the zeal of all my colleagues with whom it is a pleasure to bring such sessions to the benefit of all student participants. Dear students, let the desire to learn in you grow, and may you be passionate about your dreams. Let your imagination not be your limitation. Push yourself because no one else is going to do it for you. Great things can never come from comfort zones. I'm sure this webinar will throw light on that. Dream it. The harder you work for something, the greater you'll feel when you achieve it. Dream bigger. I sincerely hope you gain a lot from today's webinar. So keep your faith on, keep your spirits high, remain mentally active, stay tough, stay safe, stay healthy, stay blessed. Thank you. Over to you, Malay.
Audible? Yeah. Thank you, Shantini ma'am, for those ever supporting and motivating words. May I now request our principal, Dr. Anita Kanwar, to address our audience and give them a brief insights about our institution. Over to you, Anita ma'am.
Thank you, Malay sir and Shantini madam, and good afternoon to everyone. Our esteemed guest and speaker for this webinar, Dr. P.V. Bhaskar, Scientist D, National Center for Polar and Ocean Research, Ministry of Earth Sciences, Government of India. Our Vice Principal and Head of the Microbiology Department, Dr. Shantini Nair, other Microbiology Department members, all invitees, and my dear students. Warm welcome to you all from Vivekanand Education Society's College of Arts, Science and Commerce. Sir, to give you a brief introduction about our college is multi-faculty college which was established in 1979 and we are permanently affiliated to University of Mumbai. Currently, we are running 29 courses, which includes 17 undergraduate programs, 9 PG programs, and 3 PhD courses. I am proud to tell you that our microbiology department has all the three departments, UG, PG, and PhD. With the help of our very supportive and progressive management, we have very good achievements to our credit. We were accredited with A grade in our third accreditation cycle by National Accreditation and Assessment Council (NAC), and we have also received Best College Award from University of Mumbai under an urban area category. At the outset, let me congratulate and put my appreciation for microbiology department on record. The department has contributed in the college list of university toppers. Presently, in this condition of COVID-19 pandemic, where there is so much of negativity and uncertainty, department is working hard to give their students an opportunity to learn and learn from stalwarts by conducting webinars under Inspire Me series, by calling research experts from national and international level institutes. Another very important activity of the department is Microbial website. I would like to once again congratulate convener Mr. Suman Gangar for his efforts and visualization in implementing this innovative idea for benefit of not only our college students but entire nation students. I would also like to welcome and thank you, sir, from the Institute of Polar and Ocean Research, Ministry of Earth Sciences, Government of India, for taking time out from your busy schedule and telling students about the great opportunity and research facility which your institute has, and that they can also be part of this uh, facility, and if they work hard and try to achieve something in their life. Thank you, sir, and thank you, everyone. Stay safe and stay healthy.
Thank you, Anita ma'am, for your constant support, guidance, and motivation for conduct of these webinars and other programs of microbiology department. As you all know, the title of today's webinar is Indian Polar Microbiology, an overview. Polar microbiology is a promising field of research that can tell us much about the fundamental features of life. Polar microbiology addresses the adaptations and physiology of cold adapted microorganisms. It explores the ecological role that these communities play in biogeochemical cycling and examines the challenges that polar and subpolar microorganisms encounter. The biotechnology, based on polar genetics and molecular aspects, covers several key areas including enzymes, bioremediation, pharmaceuticals, and other health related applications. Indeed, bioprospecting at the poles has increased in recent years, drawing significant attention from several industrial sectors and biotech companies for commercial exploitable activities. India has enormous potential to contribute to the pursuit of scientific exploration and the preservation of polar regions. Before we further uh, proceed further into the webinar, a small announcement to all the participants. Please post your questions in the YouTube comment section. Our moderators will pick the best questions at the end of the talk for the QA session. Now I invite convener of this webinar, Dr. Shweta Patil ma'am, to formally introduce our speaker, Dr. P.V. Bhaskar. Over to you, Shweta ma'am.
Thank you, Malay sir. Am I audible? Hello, am I audible?
Yes, Shweta ma'am, you are audible.
Yeah, thank you. So, good evening, everyone. It is my privilege today to introduce Dr. P.V. Bhaskar, Scientist D, National Center for Polar and Ocean Research, Ministry of Earth Sciences, Government of India. Dr. Bhaskar specializes in microbial processes, carbon geobiochemistry, microbial food web dynamics, bacteria, phytoplankton, micro-zooplankton interactions, siderophore distribution, chemistry, and microbial diversity. His current research activities include microbial dynamics and carbon flux in the Indian Ocean sector of the Southern Ocean bacterial phytoplankton interactions in the George systems. Sir is a postdoctorate from Nagoya University, Japan, and PhD in microbiology from Goa University. He has been awarded the JSPS Fellowship by the Japanese Society for Promotion of Science. He is also the recipient of the DAAD Fellowship by German Academic Exchange Service. Dr. Bhaskar is an invited lecturer at Nagoya University, Japan. He has worked as an auxiliary investigator, University of Algarve, Portugal, and as a research associate in the Cyber Project, National Institute of Oceanography, Goa. Sir is a member of the Indian Arctic Expedition and the Southern Ocean Expedition team since 2011. He is also a member of the Scientific Steering Committee, Southern Ocean Observing Systems. Sir has a large number of publications and technical reports in renowned journals to his credit. Without much ado, let me now request Sir to begin the session. Over to you, Bhaskar Sir. Thank you.
Am I audible? Hello.
Yes,
Am I
sir.
audible?
Yes,
Yeah.
sir. Yes, sir.
So, good evening to all of you. Thank you to the Vivekanand Education Society College for this invitation, and I'm very happy to note that a very large number of students, researchers from various parts of the country are right now logged in the YouTube. So let me start my talk. So is it visible? I would like to know if the screen is visible.
Yes, sir.
Yeah. So today's talk is on the Indian polar microbiology, an overview. So the photograph that you see in this display is the National Center for Polar and Ocean Research, which is situated in Vasco, and it comes directly under the aegis of Ministry of Earth Sciences, although it's an autonomous body carrying out polar research. And we are the nodal agency for any kind of polar research activity in India. And we carry out research in the field of Arctic, Antarctic, Himalayas, and the Southern Ocean. So our director is Dr. M. Ravichandran. He's been the director for the last 3 years. And we have different research stations in the Arctic and the Antarctic. So what you can see here, this is Bharati on the top left-hand corner. This is in the Antarctic. You can see the vessel behind, which is a transshipment vessel, which brings in all the ration and the equipments and other scientific personals who stay here for the entire year. So this is India is one of those few countries which has got a permanent base in Antarctica, and our stations are manned 24/7, 12 months a year. There are very few countries who do that. Most of them have their station operational only during the summer period, and then they vacate it during the winter. But India is one of those few countries which is having 2 stations. One is Bharati, the other one is Himadri. Himadri is an older station, which is right below, along the longitude below Cape Town in South Africa. So if you take a straight transit from Cape Town towards Antarctica, you reach Himadri. Bharati is right below India. So if you take a straight route from Kanyakumari and go all the way down to Antarctica, you reach Bharati. In the Arctic, we have a station called Himadri. This is in a place called Svalbard. It's an archipelago, which is a part of Norway. and it's the northernmost station of any research activity. India occupies this station for the summer period, and we don't occupy this place during the winter. And India is also an observer for Arctic climate research. We have a Southern Ocean expedition. You can see this station, this vessel over here, which is docked about 2 kilometers off Bharati. Bharati is somewhere in the backdrop here. And this Southern Ocean expedition happens once, nowadays, for the last 2 years, it's happening once in 2 years. Earlier, it used to happen every year. We also have a station in the Himalayas. We call it Himansh. It is above Manali. You have to go beyond Manali. And here we carry out glacial studies on Himalayan glacial activity, basically to understand how glaciers are behaving vis-a-vis the climate change. And also, it's mostly geological kind of study and mostly glacial studies. Very little of biology is being carried out there. So this snapshot shows you the expeditions. The Southern Ocean expedition starts from Mauritius, and we go all the way to Antarctica, but we don't touch the land. We always carry out the research in the ocean waters, and we come back. The Arctic research, on the other hand, is carried out in a fjord. Now, fjord is nothing but when the glacial water from one end enters the valley of mountain range, and on the other side we have the open ocean. So it's like an estuary, you can say, but it's not exactly an estuary because on one side you have a glacier and the other side you have the open ocean, and you have the water, the sea, and it is having an effect of both the glacial environment and the marine environment. So this fjord is in the Svalbard. It's and here we carry out multidisciplinary studies. Actually, in all the expeditions, we have multidisciplinary studies. Whether you are a microbiologist, whether you are a zoologist, we are a marine biologist, whether you are a chemical oceanographer or a chemistry person, environmental pollution analysis, physical studies, geological studies, atmospheric studies. So practically, we have multidisciplinary approach in all this area. So polar microbiology comes under the microbial research in extreme environments, and this cartoon can highlight you what the different types of extreme environments are there. So you can see that from acid mine drainage all the way to sea ice and permafrost. So unlike the terrestrial extreme environments like deserts and arid environment or volcanoes or hyperacidic lakes, our research is mainly focused in the marine area, but that also restricted to the poles, which is close to the Antarctic and the Arctic region. So what is polar microbiology and why is it so important? So as you know that, the microbiology overall, we can differentiate into various realms, but polar microbiology addresses terrestrial microbiology, that is the soil, rocks, ice-free areas, the aquatic microbiology, which includes the polar lakes, the subglacial water bodies, the coastal fjords and marine environment, the cryosphere, which is purely the snow and ice and permafrost microbiology. The sediments means those sediments which form the bed of either the coastal waters or the lakes, and also the aerosol chemistry, aerosol microbiology. This is also carried out in polar. It also comes under polar microbiology, and also associated forms like pathogens of birds or mammals or fishes and lichens and symbionts of marine organisms which are found totally, which are restricted to polar environments. All these come under polar microbiology. So in university, we are taught that microbiology means bacteria and bacteria and bacteria. But when we do research, we find that it's not just bacteria. So anything that comes under microscope becomes part of microbiology. You can have bacteria, you can have fungi, you can have cyanobacteria, you can have viruses, you can have microscopic algae like phytoplankton, and the plankton which grows on surfaces like the rocks and the sediments. All these come under the under the title of microbiology. And if it is carried out in poles, then it is called polar microbiology. So 85% of Earth's biosphere is exposed to less than 5 degrees temperature. That means, although we consider Earth as a very warm and very conducive environment for life, if you see, 85% of the Earth's biosphere is exposed to less than 5% of 5 degrees centigrade. So 14% of this is in the polar environments, which means the Arctic and the Antarctic. I'm not including the Himalayas or the Alps or the glaciers which are in the mountains. So the Arctic region is different from Antarctic, where the Arctic is frozen ice, a frozen ocean surrounded by landmass, which is fed by rivers and land runoff. And it is defined as a region north of 65.5 degrees north, whereas the Antarctic is defined as a landmass. It's a frozen landmass which is surrounded by oceans. It has got subglacial lakes, rivers, and it is defined as a region south of 60 degrees south. So you can understand that when we say poles apart, this is how it is. They are poles apart not only in their morphology but also in their characteristics. But both are extremely cold. So both are inhabited by cold adapted microorganisms, bacteria, archaea, and eukarya. We have got very trophic microbes. We call it heterotrophs, phototrophs, aerobes, anaerobes, chemolithotrophs, chemoautotrophs, etc. So the range is extremely diverse in microbial environment, and the habitats are extremely low. They can be extremely dry, especially during winter. In Antarctica, the temperature can go down to minus 50 degrees. There is very little precipitation, so there is very little liquid water, which is very essential for life. Most of the time, it's frozen ice in the land, in Antarctic landmass. So on the top corner, you can see these pocks on the surface of snow, and you can see these channels within the ice crystals. So what this is, these are the areas where you can always find microbes growing inside these channels. So the polar microbiology is interesting because, yes, it's an extremely harsh and extreme environment, but it is very sensitive to climate change. So any small perturbation in climate can lead to a long-term effect, and it can lead to sudden changes in the environment in the poles, which will be a little difficult for the organisms to adapt to. So the organisms which are found here are pretty unique. They have to adapt to extremely dry and cold conditions. Geographically, these are isolated areas. They're not interconnected. So you will always find that many of the species are endemic to these regions. They are not found elsewhere. For example, you can find penguins in the Antarctic, but you don't find it in the Arctic. But you will find Arctic will have the polar bears, which are not found in the Antarctic. So these kind of geographically isolated environments helps to us to understand how the microbes in these environments might have evolved over a period of time in the last millions of years. What are the mechanisms? So they have cold adaptive mechanisms, and therefore they are, ideal spot for biotechnologically important molecules. And since they are susceptible to climate change, the organisms have to be very flexible to adapt to frequent any kind of drastic change in the climate, and they play a very important role in global climate, especially the microbes, because they are involved in the sequestration of carbon dioxide. They are involved in the cycling of carbon. They are involved in the carbon nitrogen cycle, sulfur cycle, ion cycle. So practically any kind of element that you find, most of it is being controlled by the organisms which are either in the oceans or in the polar caps. Now, before I go into the details, I just wanted to show you this snapshot. Like normally, whenever we do any kind of research, we first look into the microbial diversity. So we have different tools. Either you can look at either through the conventional method of growing it in labs, or you go by cell sorting, or you go by molecular mechanism of molecular identification using DNA and RNA identification techniques. Then we try to understand what these microbes are doing in the environment. That means carbon, nitrogen, phosphorus, how they are interacting with different elements, how they are interacting with different communities, whether they are within the microbial world or outside the microbial world. When I say microbial world, within the microbial world, I would say how the bacteria is interacting with viruses, how the bacteria is interacting with phytoplankton, fungi, and other organisms, and the higher organisms which feed on the bacteria. So what is their interaction? What kind of biomolecules they might be producing in order to protect themselves, or in order to, or are they parasitic? If they are parasitic, what kind of interactions they have with their host? So all these things come into all these things are then addressed, and then we need to understand whether it's a one-time process or it happens over a period of time. So we need to have sensors, we need to have instruments which are deployed in the environment, and we get continuous data for that, and then use that data for modeling. Why do we do modeling? Well, those who are doing biotechnology, even we need to model whether the output of this organism will be sustained over a period of 100 generations. If there is a reduction, dilution of the biomolecule which is being produced, then what is the rate at which it is getting diluted, and how long it will be sustained in the environment. So these things need to be modeled, and all these things have to be done. For that, we need a lot of data, and to do that, you need to have a lot of observations, and to do that, you need field sampling. So this is all interconnected. So polar microbiology in NCOR or NCPOR is a section called cryomicrobiology, which carries out research in microbiology of snow and ice. Then we have proteomics of snow and ice, a microbial community, then microbiology of the Antarctic lakes, and then evolution of genetics and biomolecules and their applications. In the Southern Ocean microbiology, we work on the diversity of the suspended matter, associated particles, associated bacterial community, different compounds like siderophores and exopolymeric substances which are produced by bacteria, biochemistry of organic carbon, interaction of microbes with the biochemistry of organic carbon, and what is happening to the food web ecosystem. And the third is the Arctic. In the Arctic, we are focusing on Kongsfjorden, where we work on the diversity, processes, and interactions. So you can see the facilities which are there available in our lab, which is self-sustained, although for high level of chemical analysis, we are dependent on other instruments through either within the institute or with other national institutes. So in the Arctic, we work in the Kongsfjorden system, the Ny-Ålesund terrestrial streams area, Arctic waters around Svalbard, and sediments in and around Kongsfjorden. And this is the archipelago of Svalbard, and this is the area where the institute is having a permanent station, Ny-Ålesund, which is a base, a small village, and India is having a station over there. And here we carry out multidisciplinary studies, whether it's microbiology, chemistry, physics, ice dynamics, glacial dynamics, atmospheric studies, practically everything which is possible. In the Southern Ocean, we carry out marine microbiology, sea ice microbiology, and microbiology in the sediments. So again, you can see this schematic representation of the Antarctic and the ocean around it. And these boundaries that you find are, which are depicted here, are the boundaries demarcated by the International Authority on Antarctic Science, and they have designated certain areas where they carry out long-term observations. And our Indian study area is somewhere along this transect, and we have a base here, and we have a base here. In the Antarctic, we carry out cryomicrobiology, lake microbiology, and environmental pollution studies. This snapshot is just to show you the interesting habitats of microbes in the polar region. So you can see this green patch in the ice. Well, you may say that it's an algae, but normally nothing is in insulation. An algal community is always associated with bacterial community. We also be associated with fungal community. So we have a diverse environment over there. And these are the algae which is attached to the base of the ice, sea ice. And these are very good source of food for higher organisms. You can also see this ice core, and you can see this brownish patch over here. This is nothing but microbes living inside the ice. And photographs you can see are photographs of aggregates or particles which are made of bacteria, algae like phytoplankton, and detritus of feeders like zooplankton, and many other microorganisms which are calcareous like foraminifera and many others. So these are the habitats you will normally find in any environment, in aquatic environment, but in polar environment you will definitely find them associated with ice and snow. So the sea ice microbes, or SIMCO, they also play a very important role. And what happens is during the poles have a very extreme climatic seasonal changes. So they have 6 months of darkness and 6 months of summer, but the actual summer is only for 2 months, when the sunlight is the maximum, insolation is highest, and then you get a lot of algal growth. And then the sun goes down, darkness, and very little sunlight, a lot of ice is formed, and the organisms which are surviving on the surface of sea, even on the terrestrial area, they get covered by snow and ice, and they start, burrowing inside the ice. They make channels inside the ice, which you can see here. There's an algae, and you can see a blue color tinge over here. That is the exopolymeric substances which are secreted by diatoms or algae, and these exopolymers are cryoprotectant. They help them to survive the winter, and you will find that along the channel they will have a lot of algal growth which appear green or brownish in color, and along with that they have a lot of bacteria. These bacteria, in association with the algae, they keep on utilizing the organic matter which is being produced by the algae, and it helps into converting the highly complex organic matter into simpler organic matter, which can be later used by bacteria. So they have the bacteria in the sea ice have extremely cold adaptive enzymes which can metabolize organic matter under extremely low temperature. And they play a very critical role in sustaining the food web during winter. So the exopolymer polysaccharides which are produced by algae are also important in formation of particles, and these particles are formation, they give rise to food for larger organisms like zooplankton, which feed on the algae and feed on the particles. The exopolymers, they kind of act like a glue, and you can see here the bluish tinge. This is an Alcian blue stained sample, and the green cells are the algal cells, and the blue color is the exopolymeric substances or exopolysaccharides which are released by the algae. And you'll find that the bacteria are closely associated with this kind of consortium. And they are very important in the marine food web, the biological pump, which is equally important for global climate. And this is just a snapshot of how an aggregate would look like in a laboratory condition, which was created in the lab. You can see these green particles. These are nothing but the algal cells which are growing in the lab, and they form clumps. And these clumps will have a lot of exopolymeric substances, which you can see here, the changes over a period of time. And we find that, the chemistry of the exopolymer can affect the kind of particles that are produced, which can then affect the kind of feeding or how much of carbon is being eaten up by the grazers. So what's being done in polar microbiology in India is very interesting. You should understand that polar microbiology started hardly 15 years, 16 years ago. So it's a very, very new field, emerging field in India. 15 years for any subject is very little, but fair amount of work has been done. And here I'm going to give you a snapshot of what is being done and what has been done. So we do carry out microbiological studies to understand the microbial diversity in the Southern Ocean. So the Southern Ocean is any water mass below the 40 degrees south. You can see these two rectangular boxes in the lower end. And what we have found is that the distribution of bacteria in the surface and the deep chlorophyll maxima and 200 meters. These are 3 different depths from where we did the sampling and we did the molecular analysis. And what we find is that urea archaeota and nano archaeota are dominant at 200 meters, which is deeper layer, which is away from sunlight. But what we find more interesting is also that cyanobacteria is restricted to the surface and the deep chlorophyll maxima depth. Now, deep chlorophyll maxima depth is that depth where the chlorophyll signal is maximum when we lower down a chlorophyll sensor. So, and later on I will show you the instrument that is being used for that. So, interestingly, we also find that proteobacteria is predominant. So whether it is Southern Ocean waters, whether it is Equatorial Indian Ocean, we do find that there is a predominance of proteobacteria and also cyanobacteria in these waters. But the archaea are more dominant in the at 200 meters and in deep chlorophyll maximum depth. And when we do a Venn diagram and we try to see, like, are these all, similar organisms which are found, or are there any unique organisms which are only restricted to certain depths? Now, you should understand that, in the water column, when we look at it, the water column, we assume that it might be well mixed. You don't expect it to be very different. But what you find is actually that at 200 meters, 28.4% of bacteria are unique. And if you see the equatorial overall, 28.4% are unique, which is restricted only to the 200 meters. So there is a distinct difference between what is there in the surface, what is there at the deeper chlorophyll maxima, and what is there at 200 meters. Now, when we break it down to Equatorial Indian Ocean waters, or we call it Southern Ocean waters, we do find that the diversity is maximum. Uniqueness is maximum in the frontal waters in the Southern Ocean, whereas in the poles, hardly 7% of the microbial community at 200 meters are very unique. Most of them are well mixed. You can find out here that 58.2% is kind of found at all the depths. So that's the difference. When you see the overall, when you club together, then you find that yes, the 200-meter community is very unique, but that uniqueness is mainly because the Equatorial Indian Ocean waters show that there is different microbial community at the surface, in the mid depths, and in the 200 meters. Whereas in the poles, it's more or less well mixed. So we also did the culturable diversity of siderophore producing bacteria, and here you can find out that we have got 16 different genera in Southern Ocean, and I think another 9 genera in the equatorial Indian Ocean waters, which we could cultivate in agar plates. And you can here also find that the dominant forms are, for example, bacilli is dominant, halomonas, which is predominant in all the 4 stations of the equatorial waters. But when you go to the Southern Ocean, bacillus is dominant, but at the same time, we also have halomonas and some other like pseudomonas, which is unique. We didn't find it in the equatorial waters, but we found more of that in the Southern Ocean waters. So what we also found was that in Southern Ocean, Saccharobacter and Rhodococcus were also dominant, which is very unique for that environment. Whereas in equatorial Indian Ocean, we also found the dominance of Pseudomonas. 50% of the, most of these organisms that we isolated were producing between 10 to 50% of siderophore. Now, what are siderophores? I have been talking about siderophore for the last 5 minutes, and siderophores are nothing but iron-binding iron-binding organic molecules. These organic molecules are produced by bacteria when the iron concentrations are very low, and these organic compounds are very specific for binding of iron. Now, it has utility. They have industrial applications. We have got pharmaceutical applications, but at the same time, they also have a role in pathogenicity. So various pathogens also produce siderophores in order to bind the iron. And this is another snapshot of how the cultural diversity is existing between the Southern Ocean and the equatorial waters. Now, this is how we carried out the isolation of siderophore producing bacteria. What you can see here is the field sampling also in the middle in the middle photographs, where the PhD student is extracting siderophores from oceanic water. The cultures are also tested for siderophore production using CAS test, Saki test, and Neelan's test, Arnold's test. And in the halo zone is considered to be positive, and non-halo zone is negative. So, what is there in the Arctic? We have been talking about what's happening in Southern Ocean, but now let's see what's happening in the Arctic. So, we have been working on microbial diversity, both in the particle-associated bacteria and its distribution. And what we here find is that, of course, these are what you can see. This is the study area on the left-hand side, and these are the stations from where we have collected the samples. And on this side, you can see the panel which shows the distribution of the relative abundance of at various level, whether it's class level, whether it's phylum level, or it's at family level. So you can find here that at group level, SAR11 clad is there, but mostly alpha proteobacteria and beta proteobacteria, they are predominant. Bacteroidetes, they are also predominant in these waters. And what controls the distribution is that we found that organic matter like carbohydrates, they control the distribution of the particle-associated bacteria. Now, why we are so interested in particle-associated bacteria is because these bacteria, which are attached to different particles, have the ability to produce more enzymes, and these enzymes can lead to dissolution of the particles, and which means that the bacteria which are attached to the enzyme, to the particles, are a good source of diverse forms of enzymes. So biotechnologically also they are interesting to understand. So proteobacteria was a predominant phylum here, and we also found that the chemistry of organic matter controls the bacterial phylogeny both in Arctic fjord and the Southern Ocean. We also work on the food web dynamics in the Kongsfjorden, and you can see this cartoon on the left. It shows that the spring season is from April to May, then peak summer is June, July, And then August, the fall starts in the month of August, and by September, the sun is below the horizon, and then it's again winter. So in this shallow, small window of 4 months of sunlight, a lot of activity happens within the fjord where the ice melts, the algae starts growing, and because of the increasing temperature, a lot of metabolic activities happen both at the microbial level, and a lot of grazing activity happens with the higher organisms coming in. This leads to the removal of carbon from the system, which goes into the deeper layer, and it's called vertical flux. And this is very important because it helps to remove the carbon dioxide from the atmosphere, and it goes into the system. And various microorganisms we find, apart from bacteria. Bacteria will be seen as a tiny dot in the environment, but when we see the other microbes, really beautiful architecture. These are the diatoms. You will have ciliates, you have dinoflagellates, so silicoflagellates, diverse kind of organisms are there. And we again we have been carrying out microbial food web studies in this region. And what we found was that, the fjord responds in two different ways. The head of the fjord, which is close to the glacier end, has a totally different food web as compared to. I don't want to go into the details of this, but all I can tell you here at this point is that the carbon flow from the microbes to the higher organisms is different at the glacial end of the fjord as compared to the mouth of the fjord. And one of the major community which supports the food web in this environment during summer are the picoplankton. The picoplankton are cyanobacteria, autotrophic picoplankton, and there are also heterotrophic picoplankton. So both of them are actually supporting the carbon requirement of bacteria as well as the higher organisms. We have been working on the interaction of bacteria with iron in the form of siderophores, and this shows the effect of pH, effect of iron on the siderophore production. And what we found is that at a pH of 8.5, which is close to the marine pH, the siderophore organisms is the maximum, whereas the siderophore production reduces as the concentration of iron goes up. Have worked out what kind of siderophores are produced by these these isolates which we grew in the lab, and we find that most of them produce syncobactin. These are dominant in various cultures. And these are also, although I'm not showing it here, but these are also found in the marine environment. When we extract the siderophore from the seawater, you also find that syncobactin is also present in these environments. So there's a good correlation between the siderophores that are being produced in the laboratory by these isolates which were actually obtained from these waters and what is being seen in the natural environment. And the identification of these siderophores was carried out using LC-MS QTOF mass spectrometry. It is time of flight mass spectrometer, quadruple time of flight mass spectrometer. And this is the way we had conducted an experiment on board the vessel to understand what happens when you have an interaction of siderophore with the plankton community. Now, I'm just giving you a snapshot to show that siderophore is very important for the growth of algae, and how it is seen here that when you add the algae with only the siderophore, there is some amount of growth. But when you add siderophore producing bacteria along with the algae, you can see the growth is there, although iron is not added in this environment. And then when iron is added, there is a significant increase in the growth of the algae. And when you add siderophore, chemical siderophore, which is obtained from the market, along with iron and algae or with the bacteria, and you find that the algal growth shoots up, which indicates that not only iron is important, but the way this iron is made available is also equally important. So the bacteria which produce siderophore are actively involved in providing the iron to the algae, and therefore it helps in the growth of the algae. In the polar biotechnology, they are extreme environments. Definitely, everybody is extremely excited to go into the biotechnological applications of biomolecules obtained from these environments. But globally, polar biotechnology is not as active as you would find in other areas like tropical environment or temperate environment. And there is a reason. See, the Antarctic comes under any kind of research in Antarctic comes under the Antarctic Treaty. And as per the Antarctic Treaty, it doesn't allow you to exploit the environment of Antarctic and its surrounding areas for commercial purpose. That's one reason why, although this area is a hotbed for biomolecules of industrial interest, the industries are not allowed to enter to do any kind of exploitation. Even if identifying a very interesting compound, you cannot go back to Antarctica to harvest or extract the organism in large amounts just to have your commercial production. So that is not allowed. And keeping in view that in mind, many of the industries have stayed away from Antarctic as far as doing any kind of commercial exploitation for biotechnologically important biomolecules are concerned. Unlike Antarctic, Arctic is much more open, and there is an overwhelming conclusion in the report submitted to the United Nations International Arctic Science Committee that R&D in relation to the biotechnology potential of Arctic, genetic resources, and the actual commercialization of such research is occurring on a significant and ongoing basis, and there is no restriction for industrial exploitation of these resources. Of looking at this is this small paper that was published from NCPOR, and you can see that the bacterial isolates which were harvested from this environment enzyme activity against diverse carbon sources, and that's why I was saying that the particle associated bacteria have diverse enzyme activity. They can produce enzyme at various levels, and you can explore them for industrial purpose. So they have diverse carbohydrate degrading cold adaptive enzymes. These sediments in the Arctic also show a lot of bacteria which produce lipase, amylase, and protease. Now, this table on the right-hand side clearly shows, actinobacteria, proteobacteria, bacilli, beta proteobacteria, gamma proteobacteria, flavobacteria. So you will come across the same group of bacteria which are found everywhere, but they are also present here, and they produce diversely interesting biomolecules or enzymes. This is another snapshot of cryoconoids. Now, cryoconoids are small holes which are formed in the soft snow wherein water accumulates. Now, wherever water accumulates, life will start progeniting. So they become a very good source of microbial life. And these are the colonies of bacteria which were isolated from these cryoconoids. And you can see this is the diverse forms of bacteria which have been identified from the cryoconoid samples. Another interesting recent biotechnologically important compound has been identified is the L-asparagines, which is obtained from the Antarctic soil and moss. Now, you can identify a compound, but you cannot go back to dig up the place and get the chemical, the organisms in large amount, which is not allowed. You can publish the paper, but you cannot go and exploit the system for commercial purpose. And this L-asparagine is interestingly found to have anti-cancer activity, and presently they are still looking into what can be, how it can be taken up at the next level, that is testing it clinical level. So globally, if you can see, the polar biotechnology is concentrated largely on the cold adaptive enzymes and life science research applications, which is taking up 34%. Then the cosmetics and nutraceuticals and food technology and animal healthcare take 10% each. Medicine and pharmaceuticals are 23%, and enzymes with industrial application is only 3%. So still the enzymes which are obtained from these environments are largely restricted to research purpose, which is in a way good for us, for researchers, may not be good for the other industries. And these are some of the companies which are already involved in carrying out biotechnological research in polar waters, and you can see that most of them are from the European mainland, or they are from the US. And mainly the reason is because proximity to the area and also the funding. So we have collaborators with CCMB, IIT Hyderabad, Burma University in Chennai, BITS Goa, and then NCL in Pune, Goa University, and Banaras Hindu University. Then also in NIO, University of Trichy, Kufos in Cochin, and MIT University in Delhi. So these are some of the collaborators with whom we coordinate, and we also have a lot of interactive research work going on. Thank you.
Thank you, Bhaskar sir, for your motivating talk and giving detailed insights about what is polar microbiology. Your talk has opened up a new field for our young researcher who will surely plan to work in this emerging area of microbiology in near future. Let us now proceed to the question answer session, which will be moderated by Dona ma'am and Shweta ma'am. Over to you, Dona ma'am and Shweta ma'am.
Good evening, sir.
Good evening.
Thank you for that wonderful talk. We have some questions from the audience.
Sure.
I'll read them out to you. The first question is by Rashmi Nene. She asks, is there any link between study of polar microbiology and astrobiology? As in both studies, we focus on extremophiles.
Yeah, definitely. There have been various meteorites which fall, which come into the Earth. Many of them land up in the poles. So they are bringing microbes from the space. Definitely, you will have a similar environment, except the fact that in space it's vacuum. But the conditions are same. It's very cold. They are exposed to extreme conditions. Here also, at the poles, the amount of UV radiations which are coming in is much higher than in the tropics because the atmosphere is thinner. If you have as we have in Antarctic, we have a persistent ozone hole, also in the Arctic. So again, the amount of UV radiations which are coming in is also high. So the microbes which are present in these in the poles may, I'm not saying because I cannot vouch for it, but logically speaking, if you say that they may have something similar to those of in astrobiology.
Thank you, sir. This question came because our earlier session was on astrobiology just before this.
Over to you, Shweta.
It's mute.
Thank you. Yeah,
Yeah.
thank you, Donna ma'am. Next question, sir. One of our student is asking, Gaurav Patekar, is asking which phylogenetic methods have been applied to compare the beta diversity of microbes in the polar regions.
Which phylogenetic methods?
Which phylogenetic methods have been applied to compare the beta diversity of microbes in the polar regions?
Means I would like to know. I would like to know to what extent he's asking the
Maybe,
method.
sir, he is trying to ask you about what type of metagenomic studies are you carrying out here, or I mean, are you doing the conventional methods, or are you doing metagenomic
Metagenomic.
analysis?
We are
Yeah.
doing metagenomic. So
Yeah.
right now we have initiated with the 16S RNA, wherein we amplify the V3, V4 region, but we can actually take it further. We initially started with V3, V4 region. Now we are planning to take it to V3 to V5, and try to also include the archaeal genomes, which can be identified. But in what aspect he's asking, I'm not very much clear when he says that which method you are using, because the methods can depend upon what you are looking for. For example, if you are very specific about the distribution of certain group only and you don't want to know about others, then you choose that kind of a method. But if you want to know the total diversity, then it will be accordingly. So
Yeah. So it will actually depend on what you are searching for, and then that tool you will be using.
yeah.
Yeah. Okay. Over to you, ma'am.
Thank you, Shweta. So the next question is by Gayatri Achari. She asks, why is industrial exploitation allowed in the Arctic poles and not in Antarctica?
For one reason is that Arctic was exploited way before Antarctic was discovered. So because you have all the countries landmass close by and you have only the Arctic in between them. If you see the map of an Arctic uh, countries, you have the Arctic Ocean in the center, and then you have the boundaries, international boundaries of US, Canada, Europe, Russia, all of them encircle the Arctic Ocean. So their natural international boundary extends into the Arctic Ocean. For example, our exclusive economic zone, which we talk about, extends into the sea along the coast. So if you see the economic zone of these countries, it will extend into the Arctic Ocean. So you cannot prevent them from, you cannot stop them from harvesting organisms for industrial purpose from within their area, because it is their country and their boundary. So it's their exclusive economic zone. But in Antarctic, there are no countries, because Antarctica is a landmass where there are no countries. So in the very beginning, they could decide that this belongs to not one country, but it belongs to the entire world, an entire global community. Since there are no inhabitants other than the penguins and the seals and the biota of that place, so there are no human inhabitants in that place. Although the southernmost tip of South America is very close to Antarctica, still they never went into Antarctica and occupied that place as a part of their, as an extension of their country. So there are no exclusive economic zones in Antarctica as compared to Arctic. So that's why in Antarctica, the international community could come out with a rule saying that, we will not allow anybody to industrially exploit this place because it's a pristine environment and we would like to protect it for science and research. And we would like to know, if you want to understand how things were there, say, 50 million years ago, you could probably get the answer in the Antarctic. You may not get it in the Arctic because probably by now people have dug up the place. But in the Antarctic, you will still get it because nobody has touched it. And that has happened mainly because of the Antarctic Treaty, which doesn't allow any industrial activity in this area.
Thank you, sir. I'm sure that has answered the question very well. Over to you, Shweta.
Yeah, yeah. Good to know, sir, that there is some place where there is a treaty and we are not going and just doing some industrial exploitation there. So the next question is from Dinoj Kutti, and he's asking, with respect to enzyme structure stabilization as a part of cold adaptation in microbes isolated from the polar region, is there any knowledge on abundance or absence of certain amino acids?
So I cannot answer this question here because this really requires a little bit of more in-depth um, study. As of now, from my knowledge, I don't know if this kind of a study has been carried out. Probably because I have not ventured too much into the microbial enzymes, because I'm not a person who works in the microbial enzymes and biomolecules that way. I'm more into the climate studies. So my interest is more in the climate studies. Although once in a while, as a spin-off of my research, I also take in some interest in biomolecules. But yeah, it's an interesting question that might be answered by someone who is actually doing uh, cold adaptive enzyme studies in the poles.
Oh, over to you, ma'am.
Yes. So there is a question by one of our alumni, Pranay Matre. He says that considering the extremophiles, especially psychrophiles, uh, what are different or special proteins present in them, like cold shock proteins or any other?
Again, so
Similar question.
now, so this is a corollary of the earlier question. Earlier was
Yes.
about enzymes. This is about structure or the cold adaptive. So normally, what we have found is that they produce different kind of polymers which protect them against the extremely dry conditions. Regarding the protein structure in these cold or presence of cold shock proteins, um, strictly speaking, it's very difficult to culture a psychrophile in order to identify those cold shock proteins. Because from a natural sample, if I want to have a cold shock protein to be identified, first I need to have some reference, and that reference has to be done in the laboratory. And psychrophiles are not easy to grow in laboratory. Practically, it's very difficult to grow in laboratory. At least I have not come across any papers where they carry out, lab control studies to understand the cold shock proteins. But yeah, what we have carried out here are those which are mesophiles, which can tolerant, psychrotolerant, and not psychrophiles. So psychrotolerance can grow at higher temperature also, and we grow them at 15 degrees and 20 degrees. But if I take a strict psychrophile, which cannot grow above 4 degrees temperature, it's not, uh, at least I have not done it, but I have not come across any reports also where they have done it to understand the cold shock proteins.
Thank you, sir.
Yeah. So the next question is from Dr. Miriam Stewart. She is a faculty at St. Xavier's College in microbiology, and she is asking, are there any specific incubation chambers and media used for isolation of these organisms? I mean, now I do understand that you are culturing psychrotolerant organisms. So what temperatures and media can be used, and how long do they typically take to grow and multiply?
Again, for example, my colleagues in Chennai, they have isolated actinobacterium. Now, they have kept it at 5 degrees or 4 degrees, and it takes months for them to get the cultures growing in a condition that they can isolate and do a molecular analysis of the isolate. Okay, so if you are strictly growing them at 5 degrees, and again, they may take 2 to 3 months to grow. Whereas what we have preferred to do is to use the psychrotolerant forms. Psychrotolerant forms we picked up mainly because of a practical restriction we had or constraint we had in the field. Because when we go in the field, we need to have, if I need to create an environment like, sub-zero condition for incubation, I need to have a system wherein I can recreate the same on board a vessel. Now, if that environment is not available on board a vessel, then automatically my preference would not be a psychrophile but a psychrotolerant, because now I would like to grow it at a slightly higher temperature. So when we do the culturing, what happens is the psychrotolerant will survive rather than the strict psychrophiles, because we are on board a vessel, we are not able to create that environment. See, one thing is there, less than 1% of marine bacteria are culturable. Less than 1%. Now, we know that how much we put on a culture plate. So when we use a serial dilution method and we use only 100 microliters to, spread plate and inoculate and incubate, from that 1% also we are taking only 1%, or maybe less than that. So it becomes very, very difficult for us to actually cultivate a psychrophile in the field, because they take 3 to 4 months to grow. And our expedition lasts for 2 months. And then the samples have to be transported in cold shipment. And that takes a month. So the problem comes in the logistics, and not in the question of technology, but in the logistics. Because once you suppose I'm able to isolate a psychrophile in the field, but by the time I transfer it to the station or to my institute, it has to undergo various temperature conditions, and by then it will not survive. So a strict psychrophile will not survive. Psychrotolerant can still survive. So even if there is a slight fluctuation in the temperature while transporting the cold shipment, like from -20, if it comes to 0 degrees also, it will not have any impact on the psychrotolerant. It can grow when you grow it back, but a psychrophile may not grow if the temperature changes. So
Yes, Dona, I'm over to you.
Yeah. So there is a question by Sabha R. She wants to know if there is any procedure to obtain polar samples for those who are working outside NCPOR.
yeah, actually, it depends upon where you are. For example, if you are a student and the college wants to collaborate, then recently we have developed the Ministry of Earth Sciences has come up with a program wherein to encourage research with other universities and other colleges. We initiate this once in 2 years, once in 3 years. Call is announced, and projects are invited from various people, various institutes. And in collaboration with NCPOR, you can go to Antarctic, you can go to Arctic, you can go to Southern Ocean. Now, Antarctic expedition is one such expedition which goes every year, and we have a call for that every year. So any institute which would like to do research in Antarctic, they can actually apply, put a proposal, and come out with a sound scientific proposal, and take and participate in this research activity. The institute, as a nodal agency, supports the logistics, supports the transport of the people, supports the stay of the personals in the Antarctic base or Arctic base. Well, I won't say Arctic, but at least in Antarctic and Southern Ocean. Everything is taken care. Right now in Arctic, we have not taken anybody else outside NCPOR and the other institutes, at least not from universities. We have got 1 or 2 people from BHU and from University of Trichy who have participated in, and also from Cochin University, who have participated in Arctic expeditions. But otherwise, many of them come from ISRO and other central institutes, the CSIR Institute or any other institute. But yes, we encourage as many proposals from participants, interested participants. The call comes out once in a year for Arctic and Antarctic expedition, and once in 2 years for Southern Ocean expeditions. So when the calls come out, they are displayed in our website, which Dr. Shweta must be knowing, our website ID. And you can go to that website, you can see the calls coming out. They are also put up in various national dailies like Times of India and Indian Express and all those national dailies. So you can actually then put up your proposal. The only thing that we don't do is that we don't fund the research. Remember, we don't fund the research. So suppose you have a funding for some research, you have the money to do the research, and you have a student with a fellowship, but you want to go and collect some samples from, say, Arctic. That can be done, but we cannot fund the research except under one scheme which has recently come up, which is called POP. Polar Opportunity for Research with other universities and other institutes. So there we invite, and the Ministry of Earth Sciences also provides funding, say between 30 to 35 lakhs over a period of 3 years, which includes fellowships for the students and procurement of small items like some consumables, and if you want to buy a laptop or a computer for your laboratory work or field work, you can buy that. Those things are provided under that scheme. But in the regular expedition of Antarctic, Arctic, and Southern Ocean, the institute provides all the logistics and management support. It is your proposal. You have the money to carry out the research. For example, you want to go to the Antarctic, you want to collect the sample, in what media you want to make that money you have to have. But to take you to Antarctic and bring you back and bring your samples back, that becomes the responsibility of the institute.
Thank you, sir. That's very encouraging to hear.
Yeah. So related to this only, sir, there is one question which has come up, that this person has a good proposal on Arctic aeromicrobiology, abundance and dynamics, and the student wants to do PhD on that project with you. I mean, especially there is requesting us to ask you this, so maybe he can go to the website and then mail you what his project is.
Yeah, he can mail me. No issues on that. And if he has a net qualification or anything, he can register for PhD under me, and we can do that. It's not a problem.
Okay, I think that is answering his question. Over to you, ma'am.
There is a question about the difficulties in this kind of research. Priyanka DeCosta says, given the difficulties and the extreme environment, would you recommend that students should go into this area of research?
If you want to do easy research, there are many other things to do. But if you want to do a research which is something different, see, it's not a question of difficult or easy. Strictly speaking, it's not like that. For example, if you are tomorrow going to work in the mining industry and you want to say what's happening in the mining area, collecting samples, getting clearances from various people, it's equally difficult. But the only advantage is that you are going back home every evening. You are going back home. But in polar, you go to Antarctic, you stay there for 2 months, 3 months, you bring the sample back. Not everybody gets a chance to go there. Remember one thing. For 1.3 billion people, there are hardly 100 people going to the Antarctic from India, and hardly 20 people going, 30 people going to the Southern Ocean, hardly 50 people going to the Arctic from a population of 1.3 billion. So if you get a chance, why not? But the question is, you are not going there for tourism. You are going there to pursue some research. So if you have a real scientific idea which you do this, and this is something that we should explore in that environment, why not? Otherwise, even collecting water sample from a dam or a river is not easy. You have to go on a boat, you have to hire a boat, you have some level of difficulties over there. But only good thing is that in the evening you go home. But here you stay in the field for 2 months or 3 months, and sometimes in Antarctic, if you are doing the wintering, you stay there for 14 months. So the entire year you spend over there. But it's not that you are in isolation. You are connected to the world. You have internet, very extremely fast internet, which you may not find out in mainland India. You get over there. So you can get cable TV. It's everything at home. You can talk to your family members every day. That's not an issue. The question is, you don't go there for that. You go there for the research. These things are added things. So if you are looking for a comfortable life, don't come.
Right, sir. I'm sure there are a lot of students who are really driven to do this kind of research. Yes, Shweta.
Yeah. So I think we'll just take the last question quickly. For culturing anaerobic bacteria, there are techniques like microculture techniques are available. Are these techniques being used for psychrophilic bacteria also?
We have not done it.
Okay.
From my side, I can tell you that much. We have not done it. And it's not something that we cannot do it. But again, the question comes, to grow a psychrophile in the laboratory, you need to have a cold room which has very strict temperature control conditions and ensure that the culture physiological characteristic doesn't change over a period of time. Otherwise, suddenly you find that what was being expressed in the nature, when you bring it back to the lab over a period of few subculturing, you lose the characteristics, and then what's the point? So these things are there. It's not that it cannot be done, but at least I have not tried it. It's an interesting thing. I can think of conducting something like that. But the major issue is logistics. To bring the culture from the field to the lab, and then to have a condition in the lab where you can grow it and sustain it. Because logistically, it takes a few lakhs of rupees to bring a small culture, small subset of culture from the field to the lab. And once it comes to the lab, if you lose it, it's a criminal waste. So we try to do with the budget we have, make sure that you don't waste samples. Otherwise, you can bring as much of samples as you want, but people are spending lakhs and lakhs of taxpayers' money. It's not somebody's personal wealth; it's taxpayers' money, and if it's not being justified, then it's a criminal waste. So yeah, it can be done. Not that it cannot be done.
Thank you, sir.
Thank you, sir.
Thank you.
Over to you, Malay, sir, so that you can continue the session.
Thank you, Bhaskar sir, for patiently answering all the questions. We will be in touch with you for the remaining questions via email if there are any.
Sure.
Thank you, Shweta ma'am and Dona ma'am, for moderating the questions and asking them on behalf of all our participants. I now request Dona ma'am to please come and propose the vote of thanks. Over to you, Dona ma'am.
Thank you, Dr. Malay. On behalf of the management of VS College of Arts, Science and Commerce, I thank Bhaskar sir for accepting our invitation and delivering an excellent talk on polar microbiology. Thank you for taking time out of your busy schedule to share your knowledge with us. It was indeed a matter of pride for us to learn about the Indian expeditions in the polar regions and about the work done by India in polar microbiology. It was an exciting as well as an eye-opening experience to know about the microorganisms living in polar regions, the ecology, and also their potential biotechnological applications. Thank you, sir, for widening our horizons and giving us a glimpse into a fascinating microbial habitat that is usually beyond our purview. I'm sure you have inspired many students to take up projects on polar microbiology. Sincere gratitude to our principal, Dr. Anita Ranwar, for her constant support. Heartfelt thanks to Mr. Shashank Joshi for the technical support he provides continuously while organizing these webinars. I also thank every member of our microbiology department for the efforts they put in to make these events successful. And a big thank you to all of you participants who have been enthusiastically participating in our webinars, and especially those who have been attending all the webinars. Thank you. Over to you, Dr. Malay.
Thank you, Dona ma'am. We have reached the end of the session. The feedback link is displayed in the description box of the YouTube channel. Kindly fill the form and give your honest feedback. Please take some time to think and answer the quiz which is coupled with the feedback form. After filling the feedback form, your e-certificates will be sent to you on your registered email ID within 7 days. The feedback link will be active for about 1 hour. Thank you, one and all. Good day. All the best.
Thank you.
Thank you, sir.
Thank you.